Alright, so I believe you have a clear idea about the CSIR NET Life Science syllabus analysis for the Unit 1 to Unit 7, which we have already talked about in the part 1 of this video series. This is the part 2 of our video series on CSIR NET Life Science complete syllabus analysis. And CSIR NET Life Science syllabus analysis regarding Unit 8 to Unit 13 will be discussed in this particular section of the video, where we talk about the CSIR NET Life Science syllabus for Unit 8 Genetics till Unit 13 biological techniques, with all the different topics name, subtopics name, and we'll also talk about the most important, moderately important and least important units with the color coding of green, yellow and red are readily available for you. If you join our coaching or get our material, you'll know that we already provide it to you, but for the first time in YouTube, complete detailed CSIR NET Life Science syllabus analysis here is for you. Let's start. Let's begin to talk about the first part, that is CSIR NET Life Science Unit 8 syllabus. And in this syllabus we have this syllabus is unedited. This is exactly from CSIR NET website. So we have Mendelian principles, and believe me, they don't ask any question from Mendelian principles. The next one is concept of gene, that is allele, multiple allele, pseudo allele, all these concepts you need to know this obviously even in the +2 level, but they won't ask any question from it. Now, green color coded extensions of Mendelian principle is most important type. Green one is the most important, yellow one is the moderately important type, red one is the least important type. So we have extension of Mendelian principle, that is a part of non-Mendelian inheritance, and codominance, incomplete dominance, pleiotropy, genomic imprinting, penetrance, expressivity, phenocopy, linkage, crossing over. And here one more topic that is not present in here is very important, that is epistasis. All are very important. The next one is gene mapping methods, that is linkage map, tetrad analysis, mapping with molecular markers. CSIR asks questions from this topic every single examinations, be it from linkage map, be it from tetrad analysis, be it from mapping with molecular markers, what not. They have asked plenty of questions from this topics only, so you need to learn this topic, gene mapping methods, very important. The next one is extra chromosomal inheritance, not that important. The inheritance of mitochondrial DNA and chloroplast DNA, maternal inheritance, not that important. Microbial genetics, that is methods of microbial gene transfer, that is horizontal gene transfer, conjugation, okay, transformation, transduction, very important. Microbial genetics important, moderately important. Human genetics, that is pedigree analysis, lodge score for linkage testing, karyotyping are there, and they are moderately important. Quantitative genetics, polygenic inheritance, heritability and measurement with QTL mapping, not that important anymore. Earlier there were questions, but not that important anymore based on previous few question papers. Mutation, molecular part of the genetics is important, like mutation, type of mutation, causes, deletion, mutation types and everything, a least important topic from them. And structure and numerical alteration of the chromosome. So the changes in the chromosome, either in the number of the chromosomes or in the shape, structure of the chromosomes, are equally important. Numerical aberrations, as well as the deletion, duplication, inversion, translocation, ploidy, polyploidy, aneuploidy, euploidy, all are super important. They belong to the moderately important topic list. So these are the topic lists. Most important, non-Mendelian inheritance, gene mapping methods, and the heritability part, that is we have the pedigree analysis and all, that is more important. And then we have like microbial genetics, structural numerical alteration, important. Like pedigree analysis problems were really more important earlier, but for past 3-4 examinations they are not asking much question from pedigree, even asking smaller questions, so we put it into the yellow colored frame. Next, so let's start with this unit 9, and first thing is the unit 9 syllabus, CSIR NET Life Science unit 9 syllabus. And they have classification biology in it, they have human like Indian subcontinents and biogeographical realms in it, the flora and fauna that are present in it. So that is also important, that's also part of it. Cladogram analysis, taxonomy analysis is another part of this unit 9. Okay, so again, like every single lecture, every single video on CSIR NET Life Science Know Your Syllabus series, we are going to color code the syllabus based on green, yellow and red. Green most important, yellow moderately important, red least important. Starting with this principle and methods of taxonomy, red least important, concepts of species and hierarchical taxa. What is taxa? The components of a taxonomic diagram, marking of that, what they mean and everything, the basics of it. The next one is level of structural organization. That is very important. Unicellular organism, colonial or multicellular form of organism, levels of organization, and everything is very, very important. Most important, levels of structural organization. Based on that, the classification for the invertebrates till phylum level and vertebrates till class level. This is very important. Outline classification of plants and animals. So again, animal classification for invertebrates till phylum, vertebrates till class. Plant classification is equally important because they have asked question from this plant classification every single time, and this is something from the hardcore classical botany topic. So obviously if you're not from that domain, you can ignore that part. And again, the animal classification is hardcore classical zoology background. So if you're not from zoology background, you can ignore that. So unit 9, I put it into our red book. Not that important because I believe that most of us who came from a modern biology background will not like reading unit 9. Natural history of Indian subcontinent is moderately important. Many people don't even give weightage to this, but in reality, if you find the question, if you check the question, you will be surprised of how many questions asked from natural history of Indian subcontinent. So this is really important. Common Indian birds, mammals, seasonality of phenology of subcontinent is really, really important. It's not that less important. Organisms of health and agriculture importance like parasites, pathogens of humans and all, not that important. So let's start. So now the question, let's talk about it. The first thing is unit 10, CSIR NET Life Science syllabus, and the syllabus starting with the environment, physical environment, biotic, abiotic factors and everything. Not that important. Although you need to know what environment is, but this is a topic from +2 level and nobody cares much about this unit anymore in CSIR NET Life Science syllabus. You need to know the basics obviously. Now, as per our rule of the discussion of these topics, we have green, yellow and red color coding. Green is most important, yellow is moderately important and red is least important. So again, starting here, we have habitat and niche, which is yellow colored, moderately important topic. What is niche? The ecological niche concept is really important. What is the difference between habitat and niche? What is fundamental niche? What is realized niche? All are equally important. Population ecology, most important, among most important. Growth curves, population regulation, metapopulation, demes, dispersal, mainland island geography, R and K selected species, everything under population ecology is super important. Species interactions, the common species interactions part is important, which is a part of the community ecology. So I should put the species interactions a part of the community ecology, which is very, very important. Then we have ecological succession, which is a part of ecosystem ecology, moderately important. We have ecosystem ecology, that ecology flow of minerals and energy, forest ecosystem, grassland ecosystem, which ecosystem is more efficient in energy efficiency. That is very, very important. We have biogeography, major terrestrial biomes, theory of island biogeography, biogeographical zones of India, why they are important. Now, although this is not that important, I can put it in the least important part, but they ask question related to the biodiversity hotspot. And that comes here, the applied ecology, environmental pollution, global warming, biodiversity status changes, the red book, IUCN list is very important. Conservation biology is also important under moderate important topics. Principle of conservation, different conservation projects that are going on in India. You need to shed lights on Indian conservation strategies that we're using, whether conserving a single organism better or a whole environment is more important. Very, very important. These are the topics. The green one most important. The red, biogeography portions and species interactions, I'll put into the red section, but rest are written in here. Now let's talk about syllabus first. So CSIR NET Unit 11 or CSIR NET Evolutionary Biology topic list. So the syllabus is kept as it is from CSIR NET website. Now in this syllabus, I placed the color coding of green means most important, yellow means moderately important and red means least important topics. So with this emergence of evolutionary thoughts, including the Lamarckism, Darwinism, origin, the concept of variation, adaptation, struggle, fitness, natural selection, all these things that play a role in the process of evolution is very, very important. Many questions are asked from this topic. And the evolutionary synthesis, the theory behind it is also important. Next is origin of cell and multicellular organisms in evolution. Now, there was a time when they ask a lot of questions from Urey Miller experiment. They ask a lot of questions from the biogenesis theory. But now, for last 3-4 CSIR NET examinations, they are not asking many questions from this topic anymore. So, we put it as a red color. Paleontology and evolutionary history, that is the evolutionary time scale, periods, epoch, major events in the time scale, including the organism, the growth of the organism, and also the extinction. All of them are equally important and that belong to yellow category. Molecular evolution, concept of natural evolution, as well as the molecular tools for phylogeny, classification, and everything that is in place. This is not that important, that they don't ask direct question from that. The mechanism is very important. The mechanism of molecular genetics is very important, and that is population genetics. So population genetics means Hardy-Weinberg principle. That is another most important part from unit 11, along with the emergence of evolutionary thoughts. Okay, so the gene pool concept, the allele frequency, gene frequency, changes of the allele frequency in a population is very important. The concept of fitness and molecular fitness, how do we calculate, how do we measure, all these things are really important. Brain behavior and evolution is another very important concept. Here we have behavioral part, that is in case of animal kingdom, kin selection, reciprocal altruism, both are equally important. Apart from that, the biological clocks, Zitzberg, as well as we have the aggressive behavior, habitat selection, all the foraging behavior. So all these things, the mating systems, territoriality, among them particularly the biogeographical, the biological clock is very important. Circadian rhythm is very important. So all these 6 different topic list in unit 11, you see the evolutionary thought, particularly the difference between the gradualism and punctuated equilibrium, that part, natural selection types, the mechanisms and involvement of population genetics is very important, and brain and behavior part is also, this is very important. And like every single video of our syllabus analysis, here I just cut and pasted the syllabus from CSIR NET website, and I'm going to give you a color coding. Green, most important topics; yellow, moderately important topics; and red, least important topics for you. Starting here with this microbial fermentation and production, not at all important. Like, we were not even counting it. That's why there's no color code out there. It's there, fermentation technology, but they don't ask question anymore from this topic. Application of immunological principles, tissue and cell culture methods for plants. Now, plant tissue culture is something is really important. Animal tissue culture is also there, but the question asked will be limited from here. Next is transgenic animal plants, molecular approaches to diagnostics and identification. So, particularly the application of recombinant DNA, the application of molecular technology or biotechnology in plant development. So plant biotechnology is a section that is out there in unit 12, and they ask a lot of questions from this topic. Okay, here transgenic animal and transgenic plant production, particularly we talk about the transformation, agrobacteria mediated gene delivery system, TI plasmid, and so many things. Genomic and its application to health and agriculture is something that is involved. Again, I'm emphasizing on the idea of genomics in terms of agriculture, in terms of plant growth, plant breeding. So mostly it lean towards plant part of the story, not the animal side of the story in case of unit 12. Next we have bioresource and use of biodiversity, not at all required. Plant breeding, again the breeding involved with the marker assisted selection, MAS, and the importance of marker assisted selection over conventional or traditional breeding methods, and also QTL mapping related to marker assisted selection. We have bioremediation and phytoremediation, which is of least importance, red color. Okay, and we have biosensors, not that important. So in this list you can see there are so many topics from which remain untouched. They don't ask question quite often from the topics. So important one is the plant agricultural biotechnology and the transgenetics out there, transgenes, production of transgenic animals and plants, plant tissue culture, animal tissue culture to an extent, and gene therapy and gene knockout, knockdown, those systems. Apart from that, marker assisted selection, plant breeding. These are important topics. 13 chapter. Now first I'll start with the syllabus of CSIR NET Life Science Unit 13, and the syllabus I just cut and pasted from CSIR NET website, but it's color graded according to my personal experience as well as 10 years question paper study. Green means most important topics, yellow means moderately important topic, and red means least important topic. Let's start with this first on molecular biology and recombinant DNA methods, that is recombinant DNA technology. Remember, molecular biology and recombinant DNA technology is RDT techniques that includes everything cloning and related to anything, cloning related anything, markers and any different kinds of markers that are utilized out there, the vectors that are utilized out there. And even after the cloning is done, then the process of screening of the libraries, DNA library, cDNA library, genomic library, everything will be there. And also different kind of markers, RAPD, RFLP, AFLP, all these markers, all these things, they are very, very important for CSIR NET Life Sciences. Remember, they're very, very important. Next one is histochemical or immunotechniques. Now, immunotechniques means related to the immunoprecipitation based or agglutination based techniques or assays. ELISA, RIA will be there, radioimmunoassays will be there, and many different types of assays will be there. Biophysical methods important, and the biophysical methods, I know that biology students have real fear for biophysical methods. I clearly demonstrated that even I think 7-8 years ago regarding the NMR spectroscopy and circular dichroism. I made videos on all this, X-ray crystallography. I've made this video on all this. I had a plan to make a new video because those videos are 7-8 years old, although the topic remains the same. You can watch that, mass spectroscopy remains the same. I'm going to make a new videos on those topics very soon. But these biophysical methods are something that you need to prepare very well. As a biology students, you fear this, but once you understand that, it becomes very crystal clear in your mind and you will be able to conquer any challenge that comes. Okay. Then we have statistical methods, moderately important statistical method, binomial Poisson distribution. Again, I have a plan to make videos on biostatistics as well. I made that I think 10 years ago. So I'll make new videos on statistical methods as well. I have a plan to make that in future as well. So again, if the time permits, I'll do that by this year end or maybe the beginning of the next year. You're going to see both biophysical techniques new videos and statistical methods new videos. Biostatistics part, particularly the chi-square test and t-test, the variance and all these concepts, you need to be very clear with. We have radio labeling techniques, which are no longer that important because radio labeling assays are no longer utilized in the lab very often. So they are kind of obsolete. Microscopic techniques are really important. I already made videos on microscopic techniques 2-3 times earlier. Now new videos are already ready. I'll be uploading that. It's already uploaded. The publishing is pending, so I'll publish that slowly at a time. The video is already ready. So microscopic techniques, you can clearly, if you stay tuned to our application, you stay tuned to our channel, you're going to see the microscopic techniques, not only theory but practical also, because I operate 2 microscopes every single day. It's my hobby to continue with that, and I can make videos on microscopy anytime, any point of time, all day long. There's no problem. But I have these videos ready. I'll share with you very soon. This is very, very important, particularly theory of microscopic techniques, that is the part related to the magnification, related to the resolution, and also bits and pieces of light microscopy, that is very important, and the difference between light and electron microscopy as well. Then electrophysiological methods are there, ECG, patch clamp technique. There are some questions like in 2-3 NET examinations, 1 patch clamp technique question may be there. So I don't recommend you to go through this particular part. Methods in field of biology, not that important, and also computational methods are out there. Now this particular part, nucleic acid protein sequence database and everything, the basics of these videos are already available earlier, like 10 years ago. You will have a separate playlist, Shomo's biology bioinformatics playlist. You can get that. New videos are already been uploaded which were not full, so I'll be uploading full videos very soon. It's already ready. So you have these computational methods or bioinformatics ready for you, and you can practice and prepare bioinformatics, the basics part of it, the basics of it and the theory of it. There's no practical aspect of it that you need to remember, but remember the most important are the molecular biology and DNA sequencing methods, recombinant DNA methods, immunological techniques methods. Second and third one is the biophysical methods. All these 3 are very, very important, and the next would be microscopic techniques and statistical methods. So if you prepare this 5 separate kinds of methods and 5 different types of techniques, you are almost ready to answer most of the question from unit 13. I always suggest that students should prepare unit 13 topics while they are preparing the respective units, because unit 13 is a combination of cell biology technique, molecular biology technique, immunology technique, right? So while you're reading cell biology, prepare the cell biology technique along with it. When you're preparing immunology, prepare immunological techniques along with it. That makes you ready. That makes you ready for unit 13 even while you're preparing other units. So if you keep unit 13 as a separated unit, it will be huge pressure on you because so many techniques are out there. I already mentioned about the name of the different techniques which are important regarding unit 13 earlier in a separate video. Every single session I put the videos and I try to present it in a different way. This time you're presenting it in a different way. So if you want to see that, if you wish to see that, in my channel, search in the unit 13 CSIR NET 13 topics, you'll find out the list, and that list will be sufficient for you. Alright, so I believe you have a clear idea about all the syllabus, all the little details of topics and subtopics that are present in CSIR NET Life Science syllabus from Unit 8 to Unit 13. If you get any benefit from this video, please hit a like button and obviously share both the videos with your friends so that they get to know about this. It's always good sharing. Sometimes you may feel like if I share important information to my friend, they'll also get to know about this, but actually it's in a public domain, so they're supposed to know it. So share it, compare, talk each other regarding this examination and utilize the positive vibe from it. Get a positive sense of competition which will help you to exceed in your CSIR NET practice sessions or CSIR NET preparation. So that's all about it. Please share the video and subscribe to this channel, and if you wish to join our coaching, the registration is going on, the details are there, you can tune in. Thank you. Bye.